a year. And this year, I think we began with a senior scientist in the field of neuroscience with a lot of years of experience and generations of students, and we conclude with one of our scientists in the field of neuroscience with the same record. Rami graduated in the Hebrew University
long time ago.
With Itzik Parnas, for those that know, and for those who don't know him. So, important name in the Israeli neuroscience field, and then moved to the NIH with the NELCON, and was recruited to Ben Gurion University as a young and energetic electrophysiologist, and since then diving with high pressure. This is like a summary of all the studies along the years.
Okay, starting now. It's very difficult to sum up about 35 years of research on high pressure, and to those who did a lot of studies with me in the early years and who are presented here, I have to disappoint them a little bit because I'm not going to present any of their data, but just about the last 10 years. But they will be acknowledged, of course. So as you see, the subject is the diving into the mystery of the brain, and the brain is always a mystery. And if you put on top of that high pressure physiology, this is a triple mystery, or quadruplet mystery, because of the technical difficulties. Nevertheless, I embarked on this about, I think 30 or 35 years ago, and I will not show data from the first about 20 years, but diving, mammals and humans, when they go into the deep sea, they are exposed to hydrostatic pressure that could come to hundreds of atmospheres in the deep ocean. But divers also, human divers, are exposed to various compressed gases that they take through the diving. And this is just the pressure values. And pressure itself is a thermodynamic variable that is expected to affect all systems. The easiest way to think about it is about activation volume of processes. And the formula is given below. The values are in ml per mole of the material, and it's dependent, of course, on the pressure. You can calculate it. But in simple terms, if a process is involved in increased volume, either of a molecule or the product of the chemical reaction, it will be inhibited by pressure, and vice versa. If it ends up in decreased volume, then pressure will enhance it. Okay? And why are we studying pressure? This is a complex question, and there are several reasons to do it. As you could see on the list, diving physiology and diving mammals and humans are very, very interesting to many people. High pressure for itself causes specific high pressure neurological syndrome that I will describe in a moment, and this is a good reason to study it when the brain actually is exposed to high pressure. And recently also long-term health deterioration are attributed to pressure exposure. Another point is the deep sea operations, either military, commercial, and so forth, of humans, and this is a good interest of commercial companies to do this type of research. And another question why people do pressure is the antagonism of anesthesia that I will not talk about it today, but it's another issue. And the last one is the question of evolutionary adaptation for life in deep oceans, especially of diving mammals, but not only necessarily not only, but any creature that lives in deep ocean in 10 kilometers is exposed to 1,000 atmospheres of pressure. So that should be very interesting. Recently, as I said, long-term effects. So, recently, not recent, the HPNS itself is a complex symptoms and phenomena. On the left, you could see that at acute moderate pressure, there is a whole list of EEG changes, and evoked potentials in the brain are decreased. Reflexes, monosynaptic is more or less is not changed. Polysynaptic are depressed, and nausea, sleep disorders, motor coordination is reduced, and cognitive performances also is impaired. If you increase the pressure to levels of about 10 or 20 atmospheres, it depends on the species, of course, you see much more severe signs and syndrome, which is the tremor, myoclonic convulsions, and vivid dreams, and also possible hallucination. And if you expose animals to high pressure, they may end up in death. Recently, long-term health effects were also attributed to pressure, especially studied on divers. And there are several clinical studies done in the US, in UK and in Norway, and the major signs are of people complain of about forgetfulness, loss of concentration, and less symptoms of decreased lung capacity, joint pains, and muscle stiffness, and so forth. And how the survey in the US, in the UK, concluded that it is not, we cannot attribute this to high pressure per se, but to other conditions during the dive. The Norwegian group attributed mostly to high pressure per se. But in comparison to HPNS, it's believed to be reversible and short-term. This is studied by many navies in the world, the Dutch, the US, and French and German navies. Nevertheless, we decided that time, long time ago, to study pressure effects directly on any preparation that we can put a hand on. And how you do it, you take a pressure chamber like on the left here, and you expose it to any preparation. Yoram Etzion will recognize this picture from his PhD study, where you can see a cerebellar slice in the chamber as seen through the porthole of the chamber. And this is the way we did the experiment. The pressure chamber is, I will show in a moment. The pressure chamber is a place where you can compress, put the preparation, put a flow, physiological solution flow, using high pressure pumps, and insert any physiological measurement apparatus inside. Everything is controlled from outside, and then we compress it with any gas we want. We use helium instead of as an imitation or substitution for hydrostatic pressure. You will see in the end of my picture, this is probably not identical, and we know it probably just last year or two. During those years, this is a long list of phenomena that we have discovered, and many of them are synaptic, I would say here, presynaptic, all this list, and this is integration on the soma, and this is the output, and this is just the action potential. I'm not going to list everything, but this is a work that all these people who are present here, like Chava, like Ariel and Yoram and others, have contributed to our knowledge. But I can sum up and say that there are three groups, major groups. One is the synaptic translation and frequency modulation. pressure affecting all various mechanisms. And the second one is the signal integration in the dendrite and in the soma, affected by any channels or conductance across these structures, and of course the intrinsic excitability and rhythmic activity that can happen in the soma. An important point is that the action potential conduction is less affected than synaptic transmission, and this is common to most all the preparation. And the preparation that we used are starting from nerve muscle preparation of invertebrate, crustacean, brain slices, isolated brainstem, spinal cord preparation like Ariel had worked on, and also lately we did oocytes and others that I will speak about it in more detail. We have
just a quick one for clarification. The brain of a diver sits in an incompressible box which is made of bones, and
אתה יכול
second...
לדבר בקול רם?
לא שומעים אותי?
אני לא.
The brain sits in a box which is not compressible, it's made of bones, and the second thing is that it's surrounded by cerebrospinal fluid which is compressible. So how do we know that if a diver is at, let's say, 60 meter depth, what his brain is exactly experiencing, his brain tissue experiences pressure compared to the outside pressure, which should be 6 atmosphere, right?
It's a good question, but your body is compressible.
Yes.
And through the fluid, the lymph and blood system, the compression is sort of transformed into the brain. That's the physical answer. Okay. So this is what we believe. Of course, minute local changes may occur, but in general, your body is exposed to the pressure that your environment, in the environment that you are in. Okay. Most of these processes that I've just described about synaptic transmission and so forth are involved the level of cytosolic calcium in the cells. And we are now, then when we realize that, we concentrated on any process that we can find and accessible to our research of what pressure does to those processes that control the calcium cytosolic level. And so we concentrated, therefore, there are many processes that control that. It's quite complex, and I'm not going to go into details for all these processes. But the major processes are the activity of voltage-dependent calcium channels, all sorts of them that you'll see in a moment, and also the NMDA receptor activity that is known to carry a lot of calcium through his current. Okay, so we concentrated on these two major things. 10 years ago, about Amir Mo, who is not here in the States, studying, specializing in medicine in the US, he started this preparation. We decided, instead of doing all this historical electrophysiological method that we brought to perfection when we put them into the pressure chamber, and all the evidence was sort of indirect. We measure synaptic level activity, we measure indirectly the currents of calcium here and there, but we wanted to study it and to show it directly what happened to those currents and to those receptor activity. So I decided at that time to move on, besides all these preparations that I mentioned before, is to oocyte preparation. It's a perfect expression system to do high pressure, because as you see, this is the oocyte of the Xenopus levis frog, relative large. It has a lot of eggs, which is called oocyte. you can dissect it out, treat it with collagenase, and then inject into the oocyte any messenger RNA that you can think of. Overexpression of this will express on the surface of the membrane of this oocyte a lot of these channels, and they will sort of mask any other minute currents that we can of course measure from the oocyte itself. And then this is a single, after 4 days incubation, you pick up one of them, put it in the pressure chamber that I just mentioned with all these technical capabilities, and that's what you see inside through the porthole here. It's again a whole stick of 2 electrodes, and this is the bath, and I don't think you can see the oocyte like a black dot there. לא רואים? אתה רואה את החץ? Oh, you don't see the arrow? Oh, I'm sorry. לא רואים את החץ, בכלל את המראה, לא רואים. לא, הוא לא מראה אותו. לא, הוא לא מראה בכלל. אוקיי, סורי. אז מה עם הפוינטר הזה, הוא יעבוד?
כן, תלחץ בבס.
זה? פה. מי שרואה את הנקודה השחורה הזו זה oocyte. שתי אלקטרודות. הפטנט הוא להכניס 2 מיקרו-אלקטרודות, ואנחנו עובדים בשיטה של 2 electrode voltage clamp. We are working a method of 2 electrode voltage clamp, which means we measure currents. Strictly currents that flow across all the oocyte and this ball of the oocyte. Okay? And we started to do it with a lot of my Ben, who is present here, that about all type of messenger RNA or voltage-dependent channels that we can put our hand on. And what marked in red, you don't see it, in red is all those that we studied. There's about 10 known calcium channels, voltage-dependent calcium channels, and the basic structure of this channel is in the middle is the alpha subunit that is alpha subunit here that pass the current, and it's all also sensor and gating and so forth, and 7 accessory subunits that are part of the full activity of the channel. And while we study them, okay, sorry, I put it in. Okay, this is just a general introduction again. All this is a neuron, and you can find different distributions of channels along this single neuron. And it's marked on this picture that all sorts of channels can be seen in various areas. So it's first point, you have to know which channel, which area that you are dealing with, and what type of calcium channel the pressure is affecting. And so our study revealed, I don't think, you can see that probably, I almost see that we studied 4 channels, the T channel, which is involved usually in rhythmic activity and pacemaking, is tremendously decreased. This is a VI curve of the channel produced by voltage clamp. The red dots are representing the pressure effect. So it's about, in this case, about 50% reduction of the current. There's no change in the voltage dependence, but just the maximum current is reduced. And on the follow just the color arrows, and the second channel that we studied was, okay, here it is the L-type calcium channel. Pressure increase its activity, increase. This is quite exceptional. It's not decreasing the current, it's increasing the current, and it's special to this channel, L-type, and I'll come to that in later on. It's already explaining some phenomena that we know about. And the PQ channel here, this is the nomenclature, classical nomenclature, is not affected by pressure, and you may remember that because it's present a lot in Purkinje cells in the cerebellum. and it's not affected, the calcium spikes there are not affected by pressure. And as I said, the only surprise that we have is the head was the N-type channel that we expected it from previous work to show tremendous decrease because it's known to be present in presynaptic terminals. And all our vast work on presynaptic terminal and synaptic activity, including neurons and others, indicated and had calcium involvement in this process. And we believe that it changes the entry of calcium into the presynaptic terminal. But in our hand, first of all, it's slightly increased, and then we added another control subunit here, the alpha 2, and it's increased quite a little, but we never observed decrease of the channel, and that's what's surprising. It's still a mystery. Probably we don't have the exact structure of the native channel. Okay, but that's science. Okay. Now, but we continued and tried to answer a more direct effect, and measurement is the cytosolic calcium level itself. In preparation, we did that in the US, as you could see, in a pressure chamber that we are looking into, we are looking into, wait a minute, in imaging system that you could see the calcium using Fura2 sensitive dye to calcium. Okay, what we have seen is that this is a major chamber built in the USA, and it's cost about $1 or $2 million just to have this major chamber, but everything is installed inside. The optical measurement, the fluorescent microscope, all the camera, CCD camera, and this is the electrophysiological path inside, all inserted under the microscope, and we are looking at the primary cortical neurons culture. This is just a general look what we saw on the microscope. Again, it's reflected outside from the chamber onto a screen. Unfortunately, this picture was supposed to demonstrate you what happened when you electrically stimulate this culture, but it doesn't work on my computer, and I had in the past a lot of problems. So imagine when you press the stimulus of the electrical stimulus in the field, the whole field now becomes red for a few seconds and then dissolve. Okay, if we zoom in now, not live, but if we zoom in into areas of this area, I marked in this is again from work PhD of Ben. We looked. I marked several cells here, and the system could choose. We can pick up any number of places of interest, and the system will follow up the change in the color. It's a ratiometric measurement of this Fura2, which is very accurate, but because of you have to stimulate to activate in two light frequencies, so it's a little bit slow. It's about 2 to 3 times per second that you can gather the information along the time axis. But nevertheless, it's very accurate, and what increased calcium is indicated towards the red color, from blue or green, which is the resting one. So this is just an example. Follow this cell, for instance, the cell in the square up there, and then after 1 second, it's lighted up. It's lighted up on the corner there even more, and this one stays for a while. But some others, you get just the flash, and they decay. If, for instance, pick up this square, the lower square, and then it's light up, and now it comes back to normal. So the response of any cell may be changing, and it depends on each cell. But when you measurement, you do an averaging of 20, 30 cells. We can measure also different dendritic area. If you look in this area in the middle, Here you see areas that became orange between the cell, and that's are dendrites, which are quite extensive in this tissue culture, and they have become also orange or red for a flesh time and then decays. So calcium also is affecting the dendrite. We don't see axons here, so it's all limited to dendrite, I guess. And this is another finding that we had through this experiment, that also during rest, without stimulation, just exposing to pressures, the resting level of the cytosolic calcium is slowly increasing, quite a lot, and it's not recovering. So there might be a problem of removal of calcium in these cells, at least under pressure. The most important thing is also the experiments were done in 3 or up to 6 atmospheres, 88. This is quite low pressure, and it was we were surprised to see that the increase of the transient after the stimulation is affected by pressure. Okay, and I said all these other things. So this is quite low pressure, and it's surprising. Sorry, this is the timing that I forgot to show you. Okay, and here I conclude the oocyte and tissue culture and of calcium channel, and I move on with a lot of work that Amir Moore, who was not here, and then Chava did synaptic, and Alice did on NMDA receptor. And this is just a general description, schematic description of the receptor, and you could see that it is quite complex. It conducts calcium out, sodium and calcium inside into the cell, embedded, and there are various sites of control, but NR1 or GLUN1, one is sensitive to glycine, this is sensitive to glutamate, and they are coagonists to the receptor. One of them is not enough to activate it, we need always 2, and as you see in the list on the left, there's a lot of and also magnesium inhibition here, and there's zinc inhibition here, possibly here inside, but they are also on NR2 side, which is quite different. There are 2 types of zinc inhibition of the receptor activity. So we decide, and the structure is interesting. It's a quadrimer, if you can see at the bottom here, and it's always the simple structure is a dimer of dimers, 2 of GLUN1 and 2 of GLUN2 subunits of the receptor. There are about 32 subunits, dimers, variations of the receptor due to 4 types of GLUN2, different gene, and 1 NR1 gene which is can go splicing, gene splicing, and 8 variants, so you can think of about 32 receptor subtypes, which is very, very, very complicated. I mean, if you are going to screen all of them, this is almost impossible. But Alice did quite a lot of work after Amir. So Amir, this is just one example of electrophysiology. This is by Amir, demonstrating important thing. This is a train of 3 stimulation in a brain slice preparation, hippocampal one, dentate, hippocampal CA1 slices, 3 stimulation, and this is the current of the receptor, the sort of inward deflection. This is a voltage measurement. Afterwards, we did current measurement with the patch clamping, macro patch, but this is the same idea. In control experiment, in this case, it was a 0.2 atmospheres, ADA, and it was with the dependence of the response, either on pressure from left to right, and on the concentration of magnesium, which is indicated on the trace. So what you could see, first of all, this is control, and now you move it to pressure, 0 magnesium in the preparation, so we could see the response, and it's not blocked, and you see how much it's potentiated compared to the control. Now see how magnesium is blocking it. At 1 millimolar, the whole response is blocked in the medium. And when under pressure, this is quite high pressure, it's 10 megapascal, it's 188. If you increase the magnesium, then you have to go up to about 3 times higher with magnesium in order to block the response. This is was a new finding at the time, and then we went on to study all types of combinations that we can think of or can be performed. We got a messenger RNA from Professor Michael Holman from Bochum, Germany, and Amir studied mostly the effect on GLUN2 activity, and Alice on the left, and Alice studied mostly the effect on GLUN1 and kept the GLUN2 constant, but note that in this case the GLUN1 was this, and the GLUN2 in this case was GLUN2A only. And Amir showed in his previous study the 2A is increased. The other is not surprisingly reduced, but Alice showed that there was a change of response. All others decreased. So the conception that NMDA receptor activity is increased during HPNS is not correct if you study single specific receptor. It's only one combination that contains, one type of combination contains GLUN2A. With all type of GLUN1, it will increase. That's what Alice showed here in this study. Everything is increased. Control, pressure, and the combination is seen here. Control, pressure, and it's reversed. So now we narrow the effect to a specific type of NMDA. We cannot speak about everything. And electrophysiology that we have done revealed several things. That on this I don't show the whole result. That saturation or aggregation are not the reason for decrease in pressure, and modification in stoichiometry also is not affected, and also the glutamate dose response curve of any of the receptors is not affected by pressure. So we were a little bit we're amazed how it's not we cannot see the mechanism for this. And just recently, it's on the figure on the left, we discovered that this receptor has a very unique inhibition by zinc. And the zinc is not voltage. This inhibition is in the nanomolar range, and it's not affected by voltage like magnesium or other zinc inhibition that blocks the pore. It's probably, the site is not on the pore, it's a little bit away. And in a set of experiments which are quite preliminary, but I think we show that the effect can be explained by that pressure is eliminating the zinc inhibition. We don't add zinc into the solution. It has a nanomolar concentration in any solution that we pick up with sodium or potassium current, and if we eliminate the zinc by using T-PEN, which is a strong chelator of the zinc, we can increase the response, just without anything, not pressure. Wow. Okay, wait a minute. Here, control, put the blocker of zinc, chelator, we increase the response by about 50% or 30%. And now we take another set of experiments This is the control. We compress it, and as we know, the response, the current are increasing by 40–50% of this combination, and then we add the T-pen, the chelator, nothing happens. So it's kind of a replacement, competition, and we believe that the pressure removes this type of inhibition. Another option for reason for increased activity was we assume that there is an increase of the number of the receptors on the membrane by pressure. This is probably found not correct by molecular biology method that you could see here. We measure by immunoblotting the number of protein that contains gluN 1A on the membrane of the oocyte after exposure to pressure compared to control, and then we do Western blotting to find out the level of the protein, and you could see after all the averaging and normalization, and this is the blotting itself, that the total is not changed, the bound is not changed, and also the bound total and the unbound is not statistically different change. This work was, of course, work with collaboration or supervision of Chava Golan, who is expert on this method than I am myself. And so this option also was rejected. No increase in the amount of the receptors on the membrane. So the only option that is left is this zinc inhibition, which we still have to work on. And recently we thought, okay, the electrophysiology and molecular biology, we have not finished with these methods, but maybe we should go into another modeling or another option that will reveal more structural details or possible more pinpoint mechanism for this increase of the receptor activity. So we went on and collaborated with Evgeny Moscovich to do a molecular dynamics simulation using what they call GROMACS software developed in Holland, I think. It's a very, very complicated. And the method is that you create a box. You see, this is kind of a cartoon of the receptor, the NTD area, the agonist binding. Oh, sorry, I forgot. Sorry, I will show you. This is a cartoon of the receptor. This is the NTD area. This is the agonist binding area. And this is the transmembrane area. The cytodolic domain is missing in this simulation because all people who did crystallography or anything structural study cut it off. They cannot have the structure with this domain at the moment. But the program idea is you take a box and you put all the structure, the membrane, which is a DOPC, sorry, it's a phospholipid double structure of a membrane, artificial membrane. It doesn't include all components of normal membrane, but it's a good simulation, and then you plant or embed the receptor protein inside this model, and it's a box contains, as you see, thousands of particles and molecules, like total 35,000 particles or more, and then the computer, you need a supercomputers to do, and you impose any models of mechanical forces and so forth between the molecules by the atom level. You calculate every atom level and each atom, what happens when you start compressing it, and you calculate it physically, and you need a supercomputer. This work was done on a Titan supercomputer in the US, run by this guy, and we get the details, and Alice did most of the analysis of this project. So what we see, this is just a graphical description of what we see, is that this is the control. This is the control. Then you apply 25 atmospheres hydrostatic pressure only. And this is what the computer comes out with. You see the membrane here is represented like with color, and red dot is the negative head. And this is what we see from the receptor. You see some changes, but they are not dramatic. But if you use helium, a strange phenomena happens. The helium is represented in gray dots. The helium molecules diffuse into the membrane between the two layers. Of course, this is exaggerated situation because the boundary conditions of this box, it's not closed. The membrane is not closed. So at the end, it opens like an X structure. I guess if we are planning to repeat it in a vesicle, you won't see this large expansion, and you see like sausages, like format. But nevertheless, the helium is going in, and it changes the membrane properties, and it affects the protein. That's the most important thing. The protein is changed, and I will skip that. We see that this just describes how the helium is distributed, and now we go to a particular site. You can now go zoom in to specific areas after you calculate what happened, and to come out with what are the possible changes in the model, of course. So on the left, you see the magnesium site. We know from other works what is the magnesium site of this protein, and now you check it under. This is control, 1 atmosphere. This is the area. This is the area inside, and the side here is where the magnesium is supposed to bind. And you see, we measure the distances in Angstrom in each condition between the 4 points known to be important for magnesium binding. And under helium condition, you see the tremendous change compared to the other two. Again, it's quite surprising. We studied also the pore surface, which is distinguished by M2 alpha subunits, alpha helixes subunit in the transmembrane domain, and this is the control. 25 atmosphere, so it's quite shrink, but in helium it's expanded a little bit again. But on both cases, in all cases, it's probably supposedly not continuous and maybe not conducting. Nevertheless, the current should be increased, and we are not sure about it. And finally, also the transmembrane domain M4, which is very important for conductance, is also changed. The green one is the control. It's only 4 alpha helixes shown from 4 subunits that compose the receptor. And now, when it is exposed to helium, see the blue one, see what happened to this alpha helix. First of all, if you try to alignment, the alignment is quite remote, so it's large distance between the control and the 25 helium atmosphere. But second point is that the alpha helix in some areas loses its structure, which is even more severe. Again, this is a computer simulation. I cannot say it's happening in reality, but it's a viable option. And the last method that we started to study recently is the marine molecular adaptation for deep diving by bioinformatic analysis. Alice, we were lucky that in the last year or so, new information was added in the literature and in the gene banks, and we could come out with the NMDA receptor sequencing in few diving mammals. As you could see the list here that we managed, Alice managed to get, we compared to human and rat, which are the non-diving animals, and then we use sperm whale, beluga whale, killer whale, winkle whale, and river dolphin, and a few others that were incomplete. And in a gluon 1, we checked gluon 1, gluon 2, and gluon 2A, and gluon 2B. And in gluon 2, 1, very little changes. The structure is conserved in all diving mammals compared to red. Okay? In the gluon 2B, we found quite a lot of, I would say, random substitution between amino acid, but again, it didn't affect the structure of the subunit. But surprisingly, or not surprisingly, say the least, gluon 2A, that again was suspicious for zinc inhibition and other stuff, is an increased activity, is affected major by 4 conserved substitution. You see, here V is substituted for L, E is substituted for B, and so for different amino acid, and the list is given here. And these changes probably affect the structure of the protein. And what we found here is just an example. We concentrated on 2 different things. This is again the predicted. Again, it's predicted by software that takes the information from the sequence of the protein and compared it to homological structure. And as you can see, the sperm whale, which is given here in blue, compared to red. Gray in all pictures, red is gray. And you see that the indication of the change is done by factor we call RMSD, and it measures the distance between the actual structure to the control, and it gives you some ratio. And you see this is the largest distortion of the transmembrane domain, which is in the sperm whale. It probably makes it a little bit more rigid or whatever, and it's not suffering so much from the pressure. And while the others are less and less and more close to the red. And the last point of this study is oh, sorry, I'm about to end. And this is the last one that we measure the zinc side, because in our simulation we have it, we couldn't do it in the molecular dynamics simulation because unfortunately we didn't have the gluon 2A at that time, only 2B, and so we did it here. And if you see on the marking here, just a moment. This is the sites where zinc is supposed to bind. Okay, it's by amine residues, asparagine residues supposed to be here. And what is the idea is that again in sperm whale there's a major change in this spot, which is seen closely here. The blue one, this is the control, this is the river dolphin, which is in the same plane. But this change, bending and journey is got into in the lumen where the zinc is supposed to be bound. And we believe that this whale, or maybe others as well, have much less zinc inhibition, and maybe that helps them to overcome pressure effect. So I'm trying to summarize. This is a lot of work of 20, 35 years and so forth, and I will do it hopefully quickly. It's in general, pressure is not giving a general effect, as we assumed years ago. It is selective, affecting several membranes, several receptors more than others. And especially you could see that on calcium channels. Sometimes the opposite effect in different channels. So you have to know which channel you are talking about and where it's located. Secondly, pressure reduces the excitatory AMPA receptor. In all receptors, AMPA is down. But it's down because of synaptic activity, not because of the receptor inhibition. This we know also. But inhibition, action potential activity is concerned in many preparation, despite the reduction in action excitatory synaptic transmission. And this happens probably because of the list of this mechanism. GABA A and glycine receptors inhibition is reduced. So it's sort of less inhibition, so you get a compensation for the less excitation. Second, the NMDA receptor, the specific subtype, which is increased activity, and it's very, very predominant in the brain. 2A and 2B are very predominant in the brain. And of course, loss of voltage dependence of zinc, it's also increasing activation in the NMDA receptor, and also the efficiency of magnesium blockade is suspected in many preparations on this site of and dendritic integration is increased, so synaptic input may start action potential more effectively due to the L-type calcium channel increase because it's present in large quantities in proximal dendrite of most of neuron. So it will boost any synaptic activity to increase larger depolarization and will activate action potential. And I think this is one of the last summary one. It's also a little bit long, but it is important to say everything. The excessive entry of calcium may be also pathogenic. This we have to know, to take into account. And atomic level resolution analysis revealed that the pressure is not affecting, high pressure is affecting the phospholipid at that range that we are working, 1 to 100 atmosphere. This is known. Many people are surprised to hear that proteins are affected. They expect hundreds of thousands of atmosphere. Recent earlier studies suggest that you need much larger pressure to change the protein. But we suggest here for the first time, this is the interaction between the phospholipid membrane and the protein itself that bound somehow there that is changing the protein activity. And the suggested area are listed here according to our finding. Now the homology structure that we showed on the diving mammals, this is real. This is real life. It's there. This is predictions, calculation. It's not reality at the moment, but this is real, and we think that the whales, or some of the deep diving whales that can go to 2,000 meters, 200 atmosphere more or less, are lacking the zinc inhibition. That we suggest that this is the major reason for increasing activity of NMDA receptor. And just to summarize, is of course, if we know more, if we know the diversity, if on an atomic level we can pinpoint or target the prevention or borrow protection, as we think, towards the system and increase safety of diving and understanding the biological world around us. And this is to conclude. Special thanks for all these people that are listed here. If you see the graduate students here, all took part in pressure studies. Ariel Tarasuk was here, Chava Golano is here, Rich Leifstein in Jerusalem, Dr. Talpala in Karolinska, Sweden. Yoram Etzon, unfortunately for him, is here. Amir Moore is in the US, as I said. Midon Gradvol is here, also teaching in Ben-Gurion University. Ben Aviner is here at the Navy recently. And Alice Blizniuk is just submitted her PhD thesis. And these are the list of all colleagues. John Candy, I owe her my first steps in pressure. She's at Stanford University, and she also donated me the precious chamber that I'm still using today. And when she fit it. And thank you all for listening.
Questions?
Yo, תמיד היה לו, you have always questions.
Yeah. So if you know now the amino acid structure of the whey, can you express it in the outside system?
We have to construct the
CDNA.
CDNA and get messenger RNA. But to tell you the truth, Alice did some substitutions, right? She replaced the amino acid from the whey that we know, this 4-1 substitution, and put it into the rat model.
In silico or in
No, it's
Computer.
in
Silico, okay.
in computer.
Experiment ways, it's a little bit different. But we didn't get the same results of distortions. You see, remember the one that I showed you of the transmembrane domain? This is the one that we studied. So we think that the additional ones that we didn't really pinpoint that are additional structures are affecting this resistance. But this is the point that we stand now. There's a lot more to study, of course. So what you say is to express the whale
Or we need to
messenger
change
RNA.
the amino acids. You can change today with the CRISPR acid. I don't know how
Look, you're asking me to do something that I'm not expert on, but Holman, this is the guy in Germany who supplied us with the NMDA receptors, he might do it. I'm sure he
because it
can.
will be
Yes, we
terrific
need
to
to
see.
this to look.
Yes,
Mhm.
but
We need to pitch this to look.
Okay, I'm coming.
we need
Can be
most.
done.
We need someone to do the
Sounds like a
experiment,
reasonable plan.
and I'm afraid
Can be
their
done.
mentor is probably retiring from the university quite soon. But maybe somewhere else, maybe in the Navy or some other places, we'll go on with this research.
You use the supercomputer. Somebody used it for you, right?
Yes.
You took many things, but I did not see PCO2 and PO. Like you treated it like a tissue without no metabolism.
Yes. Yes.
There might be changes at the mitochondrial level that might change, and they are good. You should have modeled. It's a major fault.
You are
If
absolutely
I was
right.
on
Not
board-
just CO2 and others, they are even
The
worse.
metabolic
things.
state of the
No, I will tell you. I'll tell you a secret. This is a very passive model. We don't have even electrical charge across the membrane at that model. It's taken from molecular dynamics, known boxes, reconstructed, and so forth. So our next step, because the formulas that have to deal with the electrical charge are additional. They are not included in the GROMAX software. So you have to either create additions or to calculate all extended forces on any atom because of electrical field. We couldn't calculate the conductance of the channel because there were no movement of ion. They were present in the box, but we could not calculate it.
No, I'm coming from a different.
So it's not a live membrane, I agree.
I'm coming from a different angle into the problem because you showed pure pressure and then you said helium. Helium is a
pressure.
gas, but also PO2 and PCO2 are gases and are changing
Yes,
volume.
but we didn't change it in the moment. They were not put into, they were not included in the model.
Now going back into Fred's question.
Sorry, just as that, this model cannot calculate yet the double molecule gas form, O2 and N2. You need to do special software for
I
that.
come from
It's in
respiratory now,
preparation.
so I got the message 10 minutes. Sorry, 15 minutes. Going back into Fred's comment, which is quite exciting. Do people see like osteoporotic, like, because divers are working against force, but are they changes in the microbiome?
Not that I know of about,
Because if you
After-
were saying
after
something,
diving?
that should be like microfraction.
I don't know about it, to be honest, but
If
nobody
this point is right,
tested it.
you should see some
About
change.
calcium, osteoporosis of bones, I don't know. But
I
there,
don't think
there,
there
there
is
are,
mechanical.
there are mechanical with
Force on the
decompression.
Go along
yes.
his fault, not my fault.
No, I'm saying, there, even in wells, in Woodsoul, they found in beaching wells on the shore, I mean, and they found some signs of osteoporosis in bones due to decompression bubbles inside, but not pressure per se, decompression. Okay, cool.
Because, and then there is another differential diagnosis. Diving is not our natural. For whales and seal, okay. For humans, crazy, right? So maybe it's, and then you said sleep abnormalities and all sorts of neuro, which all might be explained by post-traumatic syndrome. Why shouldn't I say that you put me into deep sea, I have post-trauma, and this is part of HPS?
I don't think 10 atmosphere exposure to human is a trauma.
No,
the record is 700 meters in chamber and 500 meters at sea. And all divers came out, recovered, and hopefully, I mean, during the pressure they have a lot of
no, but you know that there are still post-traumatic from 1973 war, right?
Could be some post-traumatic to adventure of diving. It's not simple. You have to go through stage edge. By the way, you breathe helium. Oh yeah, the point is that you breathe helium at that depth to replace nitrogen, to avoid narcosis, and of course to keep oxygen at low level to avoid oxygen toxicity. So you add a lot of helium. And if our calculation from the molecular dynamic is true, then helium itself may cause problems, not just the pressure.
Yeah, I know.
Yeah, this is shown again in the simulation. You have to test it experimentally. We have to do it. We have to do it. Again, it's probably unique to helium because we continue doing simulation on other noble gases. And there are different effects.
You can dive mice in saturated
Xenon,
oxygen.
you know, the xenon, argon, neon, and so forth. It's being calculated now, and we'll have the details soon.
אני חושבת שהריח של הפיצה אומר שאנחנו עושים את הדיון בחוץ.
Thanks, Father, for inviting me here to conclude this exciting year.
אחלה רמי. יפה מאוד, והוא ראה רק את החידושים, זה יפה מאוד.